Hey guys, Miss Cote here, and we're continuing our unit on cell communication and cell cycle. One part of the cell theory states that all cells come from pre-existing cells. So to explore how cells divide and reproduce, we're going to start looking at the cell cycle and mitosis. So let's get started. Cell division has a lot of important jobs. For a one-celled organism, cell division means reproduction of the organism since it only consists of one cell to begin with. In a multicellular organism, cell division can mean the development of a new organism from a newly fertilized cell, also called a zygote. And throughout an organism's lifetime, cell division functions in growth and repair of old or damaged cells. Just like we have a life cycle, from birth to death with all the stages in between, cells have a life cycle as well. We call this the cell cycle. This encompasses all of the stages of a cell from when it's first formed until when it divides. Most of the time when we talk about cell division, we're talking about mitosis, when one cell divides into two genetically identical daughter cells. And that's what we're focusing on in this video and chapter. There's a second type of cell division called meiosis, which will be discussed in the next unit. Meiosis is a special type of cell division that's main function is to produce the sex cells, sperm and egg cells, for sexual reproduction. But before we start into mitosis, let's review how our genetic material is organized. Remember that all of our genetic material is organized in a molecule called DNA, and DNA is packaged into structures called chromosomes. We refer to all of the DNA in an organism as the genome, and depending on the organism, the DNA can be either a circular chromosome for prokaryotes or a number of linear chromosomes for eukaryotes like us. So let's focus on eukaryotic chromosomes for now. Remember back to when we learned about the nucleus in the parts of the cell chapter. One of the main jobs of the nucleus is to store our DNA. We learned that when a cell is not dividing, the DNA is found in a structure called chromatin, a combination of DNA and proteins. It's only when the cell is dividing that the chromatin condenses or coils up into chromosomes. And for us humans, that means we have 46 chromosomes in our regular body cells, which are also referred to as somatic cells, and half the number, or 23 chromosomes, in our gametes. Before the cell divides, it needs to make a copy of its chromosomes. That way, each one of the new daughter cells will have an exact copy of the DNA. Remember, cell divides into two genetically identical cells. It's in the best interest of the cell to condense that material into chromosomes, or else it would take forever. If you think about it, trying to separate 46 pieces of tangled stringy chromatin and its copies would be a nightmare. You need to be able to identify the parts of a chromosome. This is a picture of a chromosome that has already duplicated its material. So you can say that this chromosome is made up of two sister chromatids. Each is a copy of the original chromosome. The chromatids are attached to each other by a group of proteins called a centromere. During cell division, the chromatids separate and move to opposite ends of the cell. At this point, once they separate, each individual structure, a chromatid, is now just called a chromosome. I know it can get confusing. This is something we'll practice during this unit. So to quickly go through the process of chromosome duplication and distribution, here we have one chromosome with its centromere. At some point during the cell cycle, the DNA needs to be copied. Now we have one chromosome that's made up of two sister chromatids. The DNA of each chromatid is an exact copy of the other. When the chromatids separate, now we have two chromosomes, one in each cell. The two daughter chromosomes are identical to each other and to the original parent cell. What we just went over is a very simplistic overview of mitosis, which is the division of the nucleus and the genetic material inside of it. Cell division needs to be finished off by actually dividing the rest of the cell, and that's called cytokinesis, division of the cytoplasm, and therefore all the organelles. When a cell is not dividing, it's in a phase called interphase, inter meaning between. Interphase is when a cell is growing, doing its thing, and eventually prepping to divide again. Interphase is divided into different stages. So let's take a look. This is a great visual of the cell cycle. Notice that cell division, the M or mitotic phase, is only a small portion of the life of the cell. Most of the time, about 90% of the time, the cell is in interphase. After a cell finishes dividing, it enters the first subphase of interphase called the G1 phase. G1 stands for the first gap phase, or sometimes referred to as the first growth phase. Since this is a new small cell, this stage is mainly for growth of the cell. The second subphase is the S phase. S stands for synthesis, and this is when the cell copies its DNA in preparation for cell division. We're not going to go into DNA replication now. We'll talk about that in the gene expression unit. The third subphase is the G2 phase, or second gap phase. Here the cell is finishing up the final preparations to divide. And then the cell moves into the M or mitotic phase. And after cell division, the new cell will start the cycle all over again. We haven't taken a closer look at the specifics of mitosis, so let's take a few minutes to review the phases of mitosis. But before we do, I just want to clarify something. I don't know how you learned about the stages of mitosis in your previous biology class, but through the years, students have come into AP Biology either telling me the stages of mitosis are PMAT or that it's IPMAT. Interphase is not a stage of mitosis. Mitosis is the division of the nucleus, and interphase has nothing to do with that. Interphase is when a cell is not dividing. And to further add to this, now in AP, you will sometimes hear about another stage of mitosis called prometaphase that, you guessed it, falls in between prophase and metaphase. Anyway, one of the objectives of this unit is for you to be able to look at a picture of a cell and identify what specific stage of mitosis it's in, or if it's in interphase. You have great diagrams in your book on pages 236 and 237 that not only show you these pictures, but also have bullet points that explain what's going on at each stage. First, let's start with interphase. This picture shows you G2 of interphase. Here the nucleus is intact with a nucleolus, and the chromosomes are not visible yet. The centrioles are together somewhere near the nucleus. Remember, this cell is not in mitosis yet, although it is preparing to divide. When we officially start cell division, the first phase is prophase. The nuclear envelope is still there, but the nucleolus has disappeared. The chromosomes are coiling up and becoming visible as individual chromosomes composed of two sister chromatids. They kind of look like worms. And the centrioles have started to move to opposite ends of the cell, with the spindle fibers extending between them. Remember, spindle fibers are made up of microtubules. Now we move on to the next phase called prometaphase, which is something you didn't learn about in regular biology class. This is basically an extension of prophase. Sometimes it's referred to as late prophase. Anyway, the chromosomes have become even more condensed and the nuclear envelope has disappeared. Now that there is no more nucleus, this allows the microtubules to spread into the space where the nucleus used to be. Some of these microtubules will connect to a structure in the centromere called the kinetochore. These are called kinetochore microtubules. The microtubules that do not connect are called non-kinetochore microtubules and are used for structure and support. The kinetochore microtubules will start pushing and pulling the chromosomes to line them up, which leads us to metaphase, which is the easiest phase to recognize. Basically, at this point, the kinetochore microtubules have aligned all of the chromosomes in the middle of the cell. This is called the metaphase plate. And that's all you need to know about this phase. See, I told you it was easy. Next comes anaphase. The main thing that happens in anaphase is that the sister chromatids separate. Each is now considered to be its own chromosome, and each one moves to the opposite sides of the cell. This happens through the shortening of the kinetochore microtubules. Remember, each kinetochore microtubule is connected at one end to the centromere of the chromosome, while the other end is connected to the centriole. As the microtubule shortens, the chromosomes move closer and closer to the centriole. At the same time, the non-kinetochore microtubules lengthen, making the cell more elongated. At the end of anaphase, each end of the cell will have a complete set of chromosomes. When the chromosomes reach the opposite ends of the cell, then the cell moves into the last phase of mitosis, called telophase. This is basically the opposite of prophase, where the chromosomes will uncoil and become chromatin again. The nuclear envelope returns, as well as the nucleolus, and the microtubules are broken down and disappear. Now we have our two new nuclei, each with an identical copy of the DNA. Now all that's left to do is to physically separate the cell into two distinct cells. This process is called cytokinesis. Cytokinesis is the division of the cytoplasm and the organelles. It's the final step in creating two new and identical daughter cells. However, there's a difference between cytokinesis in an animal cell versus cytokinesis in a plant cell. You can copy down this slide, but I want to talk about the differences using pictures from the book. In an animal cell, cytokinesis happens by a structure called a cleavage furrow. This is a ring of microfilaments, otherwise known as actin, where the metaphase plate was. These filaments contract and pinch in. The book uses the analogy of pulling a drawstring until the cell is split into two. In plant cells, this happens a little differently because plant cells have that cell wall that surrounds them. Vesicles that come from the Golgi travel to the middle of the plant cell where they combine and form a structure called the cell plate. Inside the vesicles are the materials needed to make a cell wall. The cell plate elongates and eventually separates the original plant cell into two new plant cells. Just a tip, you should know the structures associated with cytokinesis in both an animal cell and a plant cell. In case you'd like to see the stages of mitosis in a plant cell, your book has some nice pictures that are on this slide here, but I don't really have time to go over each one again. We'll be using plant cell pictures in our mitosis lab. So we've been discussing how eukaryotic cells divide, but you should also know how prokaryotic cells divide as well. This is a process called binary fission. It's a much simpler process than mitosis because remember, prokaryotes do not have a nucleus and only have one large circular chromosome. Looking at these processes through an evolution standpoint, binary fission came first, since prokaryotes came before eukaryotes, and over time developed into the more complicated process of mitosis. So we start with a bacterial cell, and obviously to make two new bacterial cells, we need to make a copy of the circular chromosome. The cell elongates while the chromosome is being copied. When replication is finished and each chromosome moves to the opposite end of the cell pinches in and separates the bacterial cell into two new cells. Again, each cell has identical DNA to each other and to the original parent cell. And this is where I'm going to end this video. So now that we've gone over the cell cycle and mitosis, in the next episode, I'd like to discuss how the cell cycle is regulated. That is, the controls that are in place to make sure everything runs smoothly. And of course, what happens when the cell loses control. So stay tuned for the next video, and as always, be a good human.